In this movie, we're going to discuss the basic actions of metabotropic receptors, which are also known as G-protein coupled receptors.
Metabotropic receptors differ considerably from the so-called ionotropic receptors. To illustrate these differences, let's review how ionotropic receptors, such as the nicotinic acetylcholine receptors or AMPA type of glutamate receptors, work. Shown here is the classic nicotinic acetylcholine receptor, the type found in all neuromuscular junctions. These types of receptors are called ionotropic because they directly open an ion channel in the receptor. When ACH binds to the receptor, the transmitter opens, thereby allowing for the flow of sodium and potassium through the channel, which depolarizes the cell interior.
Metabotropic receptors, in contrast, do not have a pore or ion channel. Rather, they are coupled to a G protein, which are also known as guanosine nucleotide binding proteins. The G proteins are bound to the membrane and function as molecular switches. Each protein has three subunits: the alpha subunit and a beta and gamma subunit. The beta and gamma subunits form a stable dimeric complex that is called the beta-gamma complex. In addition, the alpha subunit is bound to a guanosine diphosphate, also known as a GDP. When the GDP is bound, the protein is in an off state. Here is how the G protein gets activated and how it operates. First, the transmitter binds to the receptor, which causes the G protein to throw off the guanosine diphosphate and bind a guanosine triphosphate to the alpha subunit. Binding the GTP activates the G protein, which is now in an on state. In the on state, the alpha subunit disassociates from the beta-gamma complex. The two subunits then migrate along the membrane and cause downstream effects, as we'll show you in just a moment. These downstream effects include phosphorylation of ion channels, as well as the activation of a variety of enzymes, such as adenylate cyclase, phospholipases, or protein kinase C. One of these downstream effects is caused by the beta-gamma complex, which migrates and binds to an ion channel, causing the channel to open or close. In this case, the beta-gamma complex binds to a potassium channel, causing the channel to open, thereby hyperpolarizing the membrane. The activated alpha subunit also migrates along the membrane to activate one or another enzymatic activity. We will see one of these downstream effects of the alpha subunit in the next slide. One cascade the alpha subunit can initiate is the activation of protein kinase A, which then phosphorylates a potassium channel. The major players in this cascade are first adenylate cyclase, an enzyme embedded in the membrane. The second player is protein kinase A, so named because it is activated by cyclic AMP, which we shall see in just a moment. The kinase has two components: catalytic subunits, the parts that phosphorylate another protein, and inhibitory subunits. When the inhibitory subunits are bound to the catalytic subunits, the kinase is in an inactive state and cannot phosphorylate a substrate. Here is how the cascade works. First, the protein binds a GDP. The alpha subunit then migrates along the membrane and activates adenylate cyclase. The activated cyclase then catalyzes a reaction that converts ATP into cyclic AMP. The cyclic AMP then binds onto the inhibitory subunits of the inactive protein kinase A, the A referring to the activation by cyclic AMP. When the inhibitory subunits bind cyclic AMP, it allows the catalytic subunits to dissociate and then become active. The active catalytic subunits then phosphorylate an ion channel, a potassium channel in this case, causing the channel to close, thereby depolarizing the cell. Indeed, this is the way that adrenergic receptors in the pacemaker fibers of the heart work. The enhanced depolarization acts to speed up the heart rate. The operation of these adrenergic receptors will be described in another movie on the effects of adrenaline and acetylcholine on the heart. An important feature of metabotropic receptors is that as long as transmitter is bound to the receptor can activate additional G proteins which are also embedded in the membrane. One of the very important features of metabotropic receptors is that the effects of the activation of just one of those receptors can be greatly amplified. Those amplifications occur at several places along the transduction pathway, and I'm going to illustrate this by the activation of adenylate cyclase. The first amplification is that the activated receptor can then activate more than one G protein, and each activated G protein then activates an adenylate cyclase, which leads to the second amplification. The second amplification is that each activated adenylate cyclase can convert many molecules of ATP into many molecules of cyclic AMP. The third amplification is that the cyclic AMPs then activate many molecules of protein kinase A. And the fourth amplification is that each activated catalytic subunit of the protein kinase A can phosphorylate many ion channels. An illustration of the amplifying power of metabotropic receptors will be seen when we consider the retina, since rhodopsin, the photopigment in the retina, is actually a metabotropic receptor. Indeed, as we shall see, the absorption of just a single photon of light will lead to the closing of hundreds of sodium channels and a detectable change in perception. So we have seen how metabotropic receptors are activated, but how are the effects of the G proteins terminated? They are terminated in four major ways. The first occurs when the transmitter disassociates from the receptor, thereby terminating any additional activation of G proteins. The second is that the GTP bound to the alpha subunit has inherent GTP-ACE activity and hydrolyzes the GTP into GDP and inorganic phosphate. The hydrolysis of GDP to GDP inactivates the G protein and allows the subunits to recombine. The third concerns the inactivation of protein kinase A. Here, phosphodiesterases convert cyclic AMP into 5' AMP, which does not bind to the inhibitory subunits. When the cyclic AMP is converted into 5' AMP, it disassociates from the inhibitory subunits, and the inhibitory and catalytic subunits recombine, thereby terminating the activity of the kinase. The fourth way terminates the phosphorylation of ion channels. Here, a phosphatase dephosphorylates the channels, thereby causing the channels to revert to the state they had prior to being phosphorylated. In this case, the channels go from a closed state, when they are phosphorylated, to an open state.
So there are four major features that distinguish metabotropic from ionotropic receptors. The first is that unlike ionotropic receptors, which directly open an ion channel, metabotropic receptors are not directly coupled to an ion channel, but rather coupled to a G protein. Because the G proteins exert their effects downstream, the effects of metabotropic receptors are much slower than ionotropic receptors, which are very fast. 3. The effects of metabotropic receptors last much longer than the effects of opening an ion channel of an ionotropic receptor. And I'm going to illustrate that very vividly in the next movie. And 4. The effects of binding a transmitter to a single metabotropic receptor can be greatly amplified. There are two final points that you should be aware of. The first is that there are both ionotropic and metabotropic receptors for many, but not for all neurotransmitters. There are ionotropic receptors for the classic transmitters, glutamate, the principal excitatory transmitter in the brain, for acetylcholine, for GABA, the principal inhibitory neurotransmitter in the brain, and for glycine. There are also metabotropic receptors for glutamate, for acetylcholine, and for GABA. So these transmitters can have different effects depending upon the receptor at the synapse. Remember, it is the receptor that determines the effect, not the transmitter. There is no, however, a metabotropic receptor for glycine. It is the one exception. But some neurotransmitters operate almost exclusively, if not exclusively, via metabotropic receptors. These include serotonin, adrenaline, and dopamine. The final point concerns the locations of ionotropic and metabotropic receptors. Ionotropic receptors are located almost exclusively in the postsynaptic membrane, where they operate as fast responders to the release of transmitter from the presynaptic cell. Metabotropic receptors, however, can be located on the postsynaptic membrane, where they initiate slower but longer-lasting effects than ionotropic receptors, but they are often also located on the presynaptic terminals, where they are activated by the release of transmitter and often act to change synaptic strength. This is a feature that we have not yet considered, but will be described in the next movie dealing with aplesia, and in the lecture when we discuss how endocannabinoids, the active ingredients in marijuana, operate in the brain.